accepting our invitation for the BIU Vision Seminar. So Dr. Weiland received his BS and Masters and PhD from the University of Michigan in Biomedical Engineering and Electrical Engineering. He then joined Wilmer Ophthalmological Institute at Johns Hopkins University as a postdoc and then appointed as an assistant professor. Then he was appointed as a faculty in the Eye Institute of University of Southern California and served as a professor in ophthalmology and biomedical engineering and as the director of Biomimetic Microelectronic Systems Engineering Research Center. A few years ago, Dr. Weiland was appointed as a professor in biomedical engineering and ophthalmology and visual science in the University of Michigan, going back to his home university. He is a fellow of the American Institute of Medical and Biological Engineering, a senior member of the IEEE, the Biomedical Engineering Society, Sigma XI, and the ARVO Association. Dr. Weiland research interest is very wide. You will see soon. Taking an interdisciplinary approach, his research interest includes retinal prosthesis, neural prosthesis, electrotechnologies, retinal electrophysiology, visual plasticity, and implantable electrical system. He has a wide experience in the industry, and maybe we'll talk about it. He worked closely with Second Sight Company, leading to numerous publications and conference presentations. The Argus implant is the only retinal implant that was approved by the FDA and was recently abandoned. I had the pleasure to hear Jim in numerous talks, and I'm excited to have him here in our vision seminar to talk about electronic visual prosthesis to treat blindness. So let's all unmute ourselves and thank Jim for arriving.
Well, thank you, Yossi, and for that introduction. I want to disclose that I have worked with Second Sight in a research capacity. So what this means is I get equipment that's not generally available that allows me to do certain types of experiments that other people really can't do. But I don't have a financial interest in the company, or never had a financial interest in the company, and I was never a paid consultant for them. Nevertheless, you should consider this when I discuss the Argus 2. So I don't think this group needs this slide, but just in case there's someone who's not as familiar with vision as I think, light is focused by our cornea and lens onto the retina, which is a multi-layer structure at the back of the eye. Here's the photoreceptors. So light actually goes through the retina to the photoreceptors. Light is transduced into a neural electrical signal and is passed through the layers of the retina, and each stage does some type of signal processing and tuning of the signal. Finally, all of these axons of their ganglion cells collect to form the optic nerve, and at various points like the lateral geniculate nucleus and finally to the cortex, the visual pathway continues until we are able to perceive the world through vision. The disease that a retinal prosthesis can treat is retinitis pigmentosa and age-related macular degeneration. And that's because these cells, the intraretinal cells, are relatively spared even when there's almost incomplete degeneration of the photoreceptors. With the resurgence in cortical prosthesis, this is where an electronic implant is placed in the cortex, that opens up the possibility to treat other diseases that result in near total vision loss. These include congenital blindness, glaucoma, or diabetic retinopathy. And there have been implants, bioelectronic implants tested in visual cortex, optic nerve, and retina. So what might these diseases look like? So if this is normal vision, where you're able to see these two kids with a healthy vision, glaucoma and retinitis pigmentosa generally affect peripheral vision. Retinitis pigmentosa is more severe of these two, and whereas glaucoma can generally be treated with drops or sometimes surgery. Severe cases of glaucoma can lead to blindness, and of course, retinitis pigmentosa has no real treatment right now in general. Diabetic retinopathy can result in patchy scotomas. Age-related macular degeneration is in some ways the reverse or the opposite of retinitis pigmentosa, in that it affects the central vision. You develop a central scotoma, but maintain peripheral vision. So people with severe AMD can oftentimes maintain the ability to move around in the world using their peripheral vision, but they can't read or see in detail well because of age-related macular degeneration. So retinal prostheses have mainly been tested in retinitis pigmentosa and age-related macular degeneration. When we talk about retinal prostheses or the artificial retina, we define the devices by where the electrode array is positioned. So the electrode array is the technical part of this device that delivers electricity to the retina. And if you put it on the epiretinal side, that's on this side of the retina near the retinal ganglion cells. The subretinal electrode is placed where the photoreceptors were prior to degeneration. So you don't see photoreceptors in this diagram because there's been degeneration, and a subretinal electrode is placed here. A third position is a suprachoroidal electrode array, and that's embedded in the layers of the eye near the retina, beneath the choroid, between the choroid and the sclera, which is so that's shown in a larger view here. So that's how we define these. And then you need more than an electrode array, of course. You need supporting electronics, and that's where these different devices have the designs vary. So just a little history, because I like history. This is a patent from 1954 of a subretinal implant where there was a photocell and a single electrode. This is by Graham Tasker. I understand this was actually tested in a patient a couple of years later, but I don't have a record of that. That would be interesting to see a publication. What you see here is an implant of Giles Brindley's patient. It's an X-ray. Here's two eyes and a nose, and the mouth is down here. What catches your attention, of course, is this grid of these modules. So each one of these modules is a coil. That coil is implanted on the skull, and a wire from that coil is going to the electrode array, which sits around the occipital pole of the brain, so in the back of your head. Each one of these dots is an electrode. So this dot is connected to a wire, which is connected to this coil. Externally, what they developed was a matching helmet, which had matching an array of coils in it. So each coil in the helmet would activate one of these implanted coils, which would then deliver charge to the occipital pole. This is very crude, but it demonstrated the principle of electrical activation of the visual pathway producing the sensation of light. These days, we have more refined ways of, and frankly, regulations as to what we can put into people. And so this is the Argus 2 retinal prosthesis, which, as Yossi mentioned, was the only FDA approved retinal prosthesis, which has since been abandoned by the manufacturer. They're not making these anymore, and they're only providing limited support. In any case, this is the external system with a pair of glasses with a camera above the nose. There's a video processing unit and a transmitter that transmits to this implanted coil. You can see these straps above and below, and those wrap around the eye like a belt. This sits outside the eye on the side of the eye, and then this electrode array goes into the eye. And through a dilated pupil, you can see the 60 electrodes positioned on the retina of this patient. And that fovea is estimated to be here. So this is a well-positioned array. So here's where we sort of are really with clinically approved devices. There's 3 devices that have achieved regulatory approval, to my knowledge. The Argus 2 has FDA and CE mark, and the Alpha IMS and the Iris 2 produced by these companies have CE mark. And there's been multiple clinical trials. Prominently, the Prima device is being evaluated clinically right now, and NanoRetina, which is an Israeli company. Argus 2 was implanted in over 300 patients. There's good evidence that it helps with navigation and improved social interaction, and helped finding large objects such as plates on a table. However, visual acuity is quite low. Phosphene vision is difficult to interpret, and studies where they were claimed to be reading letters actually the people who were doing this test took more than 10 seconds, more like 20 to 30 seconds to recognize a single letter. So it's really hard to call that reading. Both of these devices are no longer available for patients. Second Sight is focusing on their Orion visual cortex prosthesis retina implant. The company which made the Alpha IMS has ceased operation. Pixium, which has the Iris 2 and a CE mark related to it, is focusing instead on the Prima subretinal implant. So these, we can ask, why do these products fail commercially? Well, they're approved for retinitis pigmentosa. This is already a small, it's not a large market to begin with. And since these devices only provided a marginal improvement in vision, that reduced the market even further. So in order for a company to continue to produce a device, you need to maintain manufacturing, you need to maintain regulatory, you need to maintain all these operations. So they need to have a certain volume of sales, and those just weren't there. The vision that was provided by these devices was just not adequate to justify implantation in most people. So we can continue to discuss this afterwards, but that's certainly one of the reasons that the companies didn't succeed commercially. So the visual prostheses, next generation devices, well, frankly, there needs to be higher resolution so that people can receive higher quality vision. And some devices that are trying to do that are a company called Nanovision. This came out of the University of California, San Diego. I don't have any data or slides on them, but it's one to keep to be aware of. The Prima device from Pixium has some nice results in a clinical study that they've published. And recently, the NanoRetina has published their in a preprint, so not peer-reviewed, but still some information on the results from the design of their implant, which is called the NR600, and some clinical results from the clinical study. The visual cortex implants, again, Second Sight is working on their Orion implant. There's a device produced by the Illinois Institute of Technology, the intracortical visual prosthesis, and then a program called Cortivis, which is being run out of Miguel Hernandez University in Spain by Eduardo Fernandez. This is not a complete list. I'll go into some details about some of these devices before I talk about current research in my lab. So here we go. The Prima is a product of the company Pixium. This really came out of Daniel Palanker's lab at Stanford. And what they've recently done is reported on 5 implant patients. And they've achieved a prosthetic. This is not exactly correct. I believe the exact number is 20/438, which is the best visual acuity that they've achieved. The device works by capturing an image with a camera through these electronics and this controller, converting that image into an infrared image, which is then projected onto the retina. And they use infrared for a number of different reasons. One, you can introduce a high intensity of infrared light into the eye and activate these infrared microphotodiodes, which are on this array here. And then by using infrared, they don't interfere with the patient's natural vision, right? So they project infrared to the back of the eye. And then in the case of AMD, like is shown here, the person still has vision in their periphery, but they don't see the infrared projection. So you have this sort of non-overlapping visual signals, which are being projected into the eye, the natural and then the infrared to serve as a way to activate the subretinal array. And here you see the subretinal array. This is an OCT scan, so that's a cross section. If you see this green line here, then you see that in cross section, and the array is sitting nicely under the macula of this patient. And again, they've at 24 months post implant, they've shown 24/60. What was not yet done with this device is that, to my knowledge, or at least not in this publication, there was no report of the patients taking these devices home and using them continuously. So this really was a clinical research study, but I expect the company has moved on to allowing that. And we hope to hear from that, hope to see that data soon. Quickly, the NR600, the innovation of the NR600 is that whereas every other device, including the Prima, has a flat array that sits adjacent to the retina, With the NR600, they have these penetrating electrodes, which are designed to go penetrate into the retina. And the expectation is that this will lead to lower stimulus thresholds because the electrodes will be adjacent to the cells that they want to, will be right next to the cells that they want to stimulate. And in the case of epiretinal implants, you can avoid activation of axons, which leads to distorted percepts simply by having the electrodes bypassing the axons. And there's as Yossi mentioned at the beginning, there's a recent bioRxiv preprint that's available that describes the operation of this device. Briefly, it uses infrared to power the device, but there's an imaging chip within a glass capsule, and all of that is implanted in the back of the eye. And so natural light is used to drive the imaging chip, which then is converted into electrical pulses, which are delivered by these electrodes in the eye. So the visual cortex prostheses, the prominent devices that are being studied here, the Orion, is really based on the Argus 2. This is Second Sight's device. It has 60 electrodes. They use a surface array. And this is what the benefit of this is proven technology. So this is robust technology that will not fail in a person, not fail. It's unlikely to fail. So it's proven through the Argus 2, and they have mature manufacturing processes. But still, with this surface electrode array, there's only this will again be a low resolution device. But so why would this succeed where the Argus failed commercially? Well, it can be applied to more types of blindness. So the notion is, let's see if we can increase the number of diseases which can be treated, and we may be able to find enough patients who are willing to get this implant, even if the benefit to them in terms of vision is quite low. A more hopeful device for high resolution vision in the cortex is the intracortical visual prosthesis. This is from, again, Phil Troik's group at IIT. And they've developed this implant, which has, each one of these modules has 16 channels, so 16 electrodes, and these can penetrate into the brain. So now, again, similar to what nanoretina is trying to do in the retina by penetrating into the retina, here you're penetrating into the cortex and getting electrodes in close proximity to the cells that you want to stimulate. And again, the hope is there that you can stimulate at higher visual acuity, which is a necessary but perhaps not sufficient condition for producing high quality artificial vision. Recently, they reported on, and this was at the Society for Neurosciences conference 2 weeks ago, they reported on implants in patients where they put 20 of these modules into a single patient. It's quite remarkable. What's also remarkable about this is the fact that this was developed largely at a university. So there was limited company involvement. Mostly this was done at a university with the supportive vendors to make the electrode array, to make the chip. But it's a pretty remarkable accomplishment that they were able to pull all this together and get this implanted into people. So how do we achieve better results versus what we're doing now? Well, one, we need better technology, and I gave you some examples of what we're doing. But really, even with the achievement of the Prima getting to 2460 or 438, that still doesn't make the person not blind. They're still blind. One definition of blindness, a widely accepted definition of blindness, is 2200 vision. So how do we get there? How do we achieve better results? So one thing we know is that there's artificial vision versus restoring vision. Someone at the beginning of while we were gathering to start the talk mentioned congenital cataracts. With cataracts, you can pretty much restore vision. Someone can have particularly in adulthood, someone can have very poor vision from a dense cataract. You can replace a lens and all of a sudden they can see again. Artificial vision will likely, from a retinal prosthesis or even a cortical visual prosthesis, will probably never be like that. It will always be artificial, but it can still be useful. But right now it needs to be stable and have adequate acuity to allow form perception. So again, them seeing something, just perceiving a letter without really thinking about it. For that to happen, we need denser arrays and wider electrode arrays so that we can get not only high acuity vision in the center, but peripheral vision for those without peripheral vision. And then to do this, one of the tools that will be necessary is we need to have computational approaches for optimizing the vision once it's optimizing the device. There's many parallel channels that can lead at least hundreds of individual electrodes, and it's likely to be impossible to adjust those individually for each patient. So we need to develop some computational modeling approaches. So I'll talk about all these things as current research in our lab. So looking at the aspect of peripheral vision. Peripheral vision is very important for mobility and orientation. So if you look at some of the devices that are on the market, you have the Argus 2, which covers only 10 degrees by 18 of the visual field, which is roughly this red rectangle. You think of the entire visual field that a person uses, and this is all you get with the Argus 2. An experimental device called polyretina, which has a larger diameter, still only covers a limited amount of the visual field if you look at it from the perspective of the whole visual field. And it's been shown that if you don't have vision outside 60 degrees, which is about out here, then you actually have decreased mobility. So if we really want to provide vision, we need to provide vision over a wider area of the retina. So we went over the Argus 2 already. So let me talk about some of the work in my lab that we're doing for a high acuity, a wide field array. So one thing we recognize that we need to have an array that to fit against the retina, we need to have an array that curves. So what we developed is a bilayer, hydrogel PDMS bilayer. So these are two different materials, and the hydrogel is responsive when it's placed in water, as shown here. If you add water to a hydrogel, essentially it swells. And that's what we show here, is that when we control the weight or the amount of the hydrogel in our material, we can produce this curved substrate. And it works as follows. So if you have a layer of PDMS on the top and the hydrogel on the bottom, and they're chemically linked, so that boundary is chemically bonded. And if one of these swells, if the hydrogel swells, then that will force a curved shape, because this bond restricts the ability of the hydrogel to swell in one direction. So it exerts a force that causes a shape, this type of shape. And when we do that in two dimensions, we show that this is the PDMS array, but when we add a hydrogel, we're able to add curvature to that. So now we've got something that's spherical and wide and can fit into the eye and take the shape of the retina. So this is just a polymer substrate. We need to add electrodes to this in order to be functional. So here's how we did that. We use standard photolithography to create a polyimide array. And this provides electrodes, conducting lines, and insulation. This is sort of your wires, right? We need to deliver electricity to a specific point, and the way we do that is we apply the electrical signal to these metal traces, but then we insulate it with polyimide, and we expose the polyimide at specific points. For the purposes of this demonstration device, we only included 7 electrodes, 1 in the center and 1 on each of these arms. We then sandwich this in polyimide. So you may ask, why don't we pattern the metal directly on polyimide? On PDMS. And the answer is it's very challenging to work with PDMS. It's a very kind of sticky, gooey type of substance. And there are labs around the world, specifically Stephanie LaCour in Switzerland, and EPFL has achieved some success doing this, but still it's challenging. So what we chose to do is instead use the PDMS as more of a mechanical substrate. This was a very narrow and thin polyimide layer, and we made a sandwich of that. Now this has gold electrodes, so what we did is we deposited a platinum iridium on the material on this in order to allow this to be robust for electrical stimulation. This is a cross-section of the process where we show where we've got PDMS. We make these 2 different layers of PDMS, one which is solid, one with openings. And this was a non-optimal process. We actually had to mechanically go in and scrape out some of the PDS. So these were fairly large features. We then transfer our polyimide array onto the solid layer of PDMS, and then sandwich and bond these 2 layers together. And when we get rid of the silicon wafer on the top and bottom, we're left with this device. There's room, we just pushed forward in spite of the deficiencies in some parts of our process in order to get the final device that we wanted, but there's certainly room for improvement. So what we did then to characterize this device is we looked at the effect of sequential treatments on the electrodes. So there was the naive electrodes without any treatment. There's the electrodes after platinum iridium deposition, and then the electrodes after we coated the hydrogel. Right. So we needed that PDMS layer in order to bond with the hydrogel, but so that's why we followed with a PDMS layer on top of the polyimide. And what we found is that so if you look at before electroplating, there was this huge variability, and we attribute this to some lack of cleanliness of the gold electrodes, and also the PDMS contamination on the electrodes. So what we see after electroplating, which is this data here, we've reduced not only the impedance overall, but we've reduced the variability. So electroplating had a cleaning effect. Adding a hydrogel layer on top of the electrodes, we were worried that would effectively insulate the electrodes, but hydrogel actually has a reasonable conductivity, so that only increased the impedance of the electrodes a small bit. And this shows some impedance spectra of in two individual cases. Charge storage capacity is an important factor for stimulating electrodes, and we've shown, without going into too much detail, that the purple line is before electroplating. After electroplating, we have this larger charge storage capacity, and adding the hydrogel reduces the charge storage capacity, but still it's significantly improved over the naive electrode. Okay, so we've got this giant electrode. This is actually about 30 millimeters wide. How do we get this in the eye? Well, because it's flexible, we can roll it up. And that's what we show here. This is still too wide. This is about 8 millimeters wide, and that results in a large sclerotomy in the eye, as you'll see on the next slide, and when I show a video. But again, this is not optimized. And we think if we really pushed forward just to demonstrate this approach, we think by optimizing the mechanical design, by optimizing the folding strategy we use, that we can reduce this significantly, perhaps even by half. So we show you here that with a 34 millimeter wide electrode, and what does that mean? That means from this tip to this tip, it's 34 millimeters. We were able to get this through a 12 millimeter sclerotomy in the eye, which is still too large if you ask surgeons. But really, we've reduced. It's the ratio is better compared to what's done with the Argus 2, which has about a 5 millimeter wide array and requires a 5 millimeter wide sclerotomy because it doesn't fold. So here is a video. I hope this works over Zoom. So this is done in a nucleated pig eye. So we didn't put this in a person or a live animal. So just to demonstrate the approach. So this is Dr. Benjamin Young, who's the surgeon doing this. And overall, this is the work of Maru Zhou, a student who just graduated from my lab, shown here. And this is Dr. Young, who's a fellow in a vitreoretinal surgery fellow at the Kellogg Eye Center. So one thing we put in a carotid prosthesis. So this is an artificial cornea so that we could see to the back of the eye. And here we are making a very large sclerotomy, which would probably be unacceptable in a patient. But through that, he's able to insert this array. And it's dry, but once it gets in the liquid environment, the hydrogel will begin to swell. It's also been wrapped with a dissolvable material, polyvinyl alcohol, to hold it in place. And he's going to suture it closed. And there it is. And so after he completed this, he cut the top of the eye off. And it's hard to see, but we've traced out the edges of the array. So the array, in fact, actually did unfold in the eye. So obviously a long way to go with this, but we've fabricated a device that has multiple materials. It uses a hydrogel approach to unfold in the eye. So it has a functional polymer, and that hydrogel does not seem to compromise the electrical properties of the array in a way that would prevent current from reaching the retina. So we think this is a good approach to achieving peripheral vision. So again, we only included 7 electrodes here in this device. 1, 2, 3, 4, 5, 6, and then one in the center. You could imagine having a sparse array in the periphery and a dense array in the center as a way of mimicking natural vision, where we use the periphery for guidance. So this is the summary of that device. Again, if you put an electrode at this tip and this tip, which we didn't do in this device, but there's no reason you can't do it technically, we would cover 113 degrees of visual field. And again, this makes the assumption that we can provide sparse stimulation out here with just a few electrodes, which could serve to cue the person to look, and then use their higher acuity central array to see things in more detail. Intraretinal stimulation.
Can I ask something?
Yes, go ahead.
Or you prefer at the end? Is it okay?
Can you repeat the question, please?
Okay, so if you can go back one slide. So in case that you are successful with this, how do you plan to have stimulation of the, let's say, hundreds of electrodes? Is it going to be through wires or photovoltaic? What would be the source of current in this kind of a device? What are the options?
Right, so the options would be having an Argus 2 type arrangement with a can on the outside, yet however, that's really prone to adverse events. You know, we did the FDA, the United States Food and Drug Administration sponsored a seminar recently, and they asked me to look into clinical evidence for safety. And what's clear is that if you have external extraocular components, that's a strong driver for adverse events, such as breakdown of the conjunctiva and hypotony, so that's loss of eye pressure. So really this, the approach taken by the Prima and now the NR600 is the right way to go to try and get everything inside the eye. So you could imagine a photovoltaic or some type of NR600 type device sitting here or peripherally, right? So their ability to make electronics smaller, and one of the innovations from the Prima that I think is really potentially impactful is their hermetic coating technology. So they managed to show that they could coat a silicon chip in a way that's robust, at least in the in vitro lifetime testing that they did. And I think that's really going to have an impact down the road, so that we can avoid these large electronic modules, if we use an enclosure. But that's a detail we can get into later. So I would imagine something inside the eye would be the way to go, just because getting routing hundreds of signals through a sclerotomy is going to be challenging, and then the system sitting out here is going to be large, and okay. So intraretinal stimulation, we feel, is one way towards high spatial resolution. It makes sense that if you have an electrode close to your target cells, that you can get an electrode into the retina, that you can use very small amount of energy to stimulate a small group of cells. Challenges are that there's extreme connectivity in the retina, so even if you stimulate one bipolar cell or a group of bipolar cells, that signal through lateral connections could spread out. So that will be a challenge to this approach, but I think this is a challenge that we hopefully can address or solve in some way. So carbon fiber electrodes is a technical approach to that, and that's what's shown in both this diagram and these electron micrographs here. And this is one of my colleague at Michigan, Dr. Cindy Chestik, has advanced this for neural recording. And then we deposit on our platinum iridium material. I also want to disclose I have a commercial interest in platinum iridium, so I'll talk about that a bit in the next few slides. On this, in order to because carbon itself is not robust for long-term electrical stimulation. So as you can see, we have a few design choices we can achieve with with carbon fiber electrodes, including something as small as 10 microns, though the robustness of this is somewhat questionable if you try and insert something with a small tip like this. So what we did here to start looking at this approach is we had an 8-channel array of carbon fibers, all coated with platinum iridium. We, in an in vitro setup shown here, we inserted those carbon fibers into the retina. And these retinas a few weeks prior had been treated with the mouse that we injected with the GCAMP virus. So now the retinal ganglion cells, because of the action of the virus, these retinal ganglion cells will now be fluorescent, and that fluorescence will change based on the intracellular concentration of calcium. So that allows us to electrically stimulate and then record from literally hundreds of retinal ganglion cells by measuring their fluorescence. What's shown here is the view through the inverted microscope of the tips of the carbon fibers as they were inserted into the retina. There's a mouse retina in this dish. You really can't see it, but this is the holder for the carbon fiber array. So using these electrodes, we had a 100 micron electrode. So what I mean by that is that the exposed length of the electrode is 100 microns. So that's what that term means. We're able to show that when we have the electrode tip 20 microns from the retinal ganglion cell layer, we get a response with 2 microamps of current, which is quite low. And it increases in a way that suggests we're not activating axons. Though I will say in some cases in this position, we did see axonal activation. At 70 micron distance, it requires more current in order to generate similar responses. So this is the measurement of the threshold then, and we show that by being closer to the retinal ganglion cells, you achieve lower threshold. So in terms of electrode size, we compared the 50 micron exposure. Again, that's the length of the exposed electrode of the carbon fiber versus 100 micron. And as you would expect that because the 50 micron electrode for a given current has a higher current density and charge density, that required less current overall. So you can see that is shown here. And then interestingly, this was an interesting finding that was repeated several times, though I can't say it's generally our result, but it's intriguing to note that with the intraretinal stimulation using a 10 micron electrode, so that's this one, this very small electrode, we inserted that into the retina as well. We could achieve a single retinal ganglion cell resolution. So you can see that this approach has the potential to really improve the stimulation resolution. Now, will that translate into better visual acuity? You know, you still need to control your stimulation and optimize the signals you're generating, but I think it's a necessary step. You need to be able to activate the visual system at just a small group of cells, if there is a hope that we'll get high resolution artificial vision from these from retinal implants. So this is an insertion test. As you may have noticed, we inserted in the in vitro preparation because of our current measurement system. We inserted the carbon fibers from the subretinal side, but if we want to insert from the epiretinal side, as in the NR600 device, that may be more challenging because of the nerve fiber layer. Here we show that in this photomicrograph, here we're dimpling the retina, and after we advance the carbon fiber, it appears the electrodes have inserted into the retina. More evidence for that is the biological material we saw afterwards on the carbon fiber electrodes, as well as the change in the voltage transient. So in the liquid, for a given current, we developed this voltage, is shown in red. But once we were touching the retina or inside the retina, the voltage increased. So that indicates that we have a higher resistance to current flow, which would be the case because the resistivity of the retina is higher than the surrounding liquid. So altogether, this indicates that at least 4 carbon fibers can penetrate into the retina. Now, will that occur if we have 100 carbon fibers in an array and pushing into the retina? I suspect that over time, it will insert into the retina, but will it do so in a stable way that doesn't result in too much damage? And that's to be determined. In primate retina will be a more significant challenge because the nerve fiber layer is thicker and the retinal ganglion cell layer, especially around the fovea, it has sort of a non-uniform shape, let's say. as you most know. So getting, again, penetrating this array in that region will be a more significant challenge, but this is a good start. I'll probably skip this to kind of go on to the patient-specific model. We're modeling this, and our model results are consistent with our experimental findings. So we show the intraretinal stimulation has the potential to achieve better spatial resolution, and that these carbon fiber electrodes coated with platinum iridium may be a viable technology. And again, I have a commercial interest in this material. And we can we need to optimize the design factors for this array as how much we want the electrode array to penetrate in the retina, what's the smallest electrode size we could achieve. And then we can, we expect that if we were to activate multiple channels simultaneously, that would result in just a widespread activation of the retina, but by interleaving stimulation, that we can achieve resolution. We can activate small groups of cells that will be perceived as small spots of light. So the last thing I want to talk about is a recently published work where we get at the problem of what's an approach that we can use to optimize the implants programming once it's in the patient. So what do I mean by that? So these devices have independent stimulation channels. Each one of them has a number of parameters that can be modified in order to achieve the best result. And it's not clear that those modifications and those settings, those parameters, stimulus parameter settings, are the same for each channel, even in the same person. In fact, it's unlikely that that's the case. So what you want is patient-specific settings. And this has been done in other bioelectronic implants, such as spinal cord stimulation. It's been done in deep brain stimulation. There's been work showing that patient-specific modeling can support clinical decision-making in those areas. So we thought we would try this for the Argus 2 implant. So this shows an OCT cross-section of the multi-electrode array and the retina in one patient. So the approach we took was on a patient-specific basis. So we took a patient's data, which shows this is one Argus 2 patient. Here's again, the OCT cross-section, and we segmented this into the electrode array, which is shown here in this sort of bluish green, the retina, which is green, and then this fibrotic tissue growth, which forms, again, on a patient-specific basis. In this case, this fibrotic tissue growth was formed here. So all of these were modeled as different domains for our electric field model, and this tissue growth is significant because about 50% of Argus patients develop this, per a study by Stanislaw Rizzo of only 20 patients. So it wasn't a huge cross section, but still, it's something that we see quite typically. So we first use the finite element method to get a voltage gradient. So what is this we're looking at? So this is the eye. The dark blue is the eye. This royal blue shows the Argus electrode array, and then the red hot spot here is the electrode we've activated. And then these are your field lines from that electrode back to the return electrode, which is this the case which sits outside the eye. We eventually determined that it wasn't necessary to model this case, that we could just set the ground at infinity, but in this publication, we actually modeled the case. And then we solved the field equations for this. So this is physics. Once we have those voltages, then we apply those to our biophysical model, where this is our neuron model of the retinal ganglion cell, with these different areas, which have all shown to be somewhat significant. In particular, the sodium channel band is generally the area of activation for an electrical stimulation of retinal ganglion cells. This is the equation that was used, and that's what's shown here. Now let's talk about these diagrams. This is where the electrode is sitting on the retina. And this is a model of the trajectory of retinal ganglion cell axons. So it's not like spokes of a wheel or ray. It doesn't project out uniformly. It forms this shape, and it does so because in primate, The fovea is about here, and these fibers are trying to avoid the fovea so that the fovea can get less light scatter. So they have this arcuate pattern. So we modeled that. And then this is a single electrode array. And then we have, I think, 400 or maybe 200 retinal ganglion cells, which you can sort of make out here. There's a small little dot, which is your retinal ganglion cell soma. And from that dot, there is a line, which is the axon. So it's a three-dimensional model of hundreds of retinal ganglion cells in proximity to a electrode. And the trajectory of the axon is based on where this electrode is in this patient. And then the other aspects of this are the presence of the nerve, of the retinal, of the connective tissue in this model. So we show that we get a, again, that we see how our biophysical model is responding. And we see that the sodium channel band, SOCB, is the most sensitive area to electrical stimulation. That's consistent with experimental results. We measured threshold in this patient. So again, we had the benefit of using modeling of the device in a patient and then going to that patient and measuring the perceptual threshold. So we could actually get data from the patient. And this is what we determined. This is their threshold map. We measured 30 electrodes. And then thresholds is determined by fitting this function to threshold data. And then at 50% response rate is what we consider to be threshold, which is right here then. And these are the results. So we did linear regression showing that we have a significant correlation with the patient reported threshold. So this is our model prediction. This is our patient reported threshold. And when we look at our results improve, when we only look at thresholds above 100 microamps. And the justification for doing this is that lower numbers this may be a result of a high false positive rate. This may not be true positives in this low regime. And then we look at with the testing block with the highest thresholds, which is the red block. We had an even higher correlation value, even though our p-value suffered because the number of samples we have is lower. So overall, we show that our patient-specific model can match the estimated thresholds that the patient reports. And this is important because each one of these thresholds takes about 20 minutes to achieve. So if you multiply that times 60, you're talking about hours of time the patient's going to spend saying yes, no, and that can get very tiring and frustrating. And that's with only 60 electrodes. So you can imagine if you have hundreds of electrodes that you're trying to tune, wouldn't it be better if you could take a picture of the patient's eye with OCT, create a model, and then make a program for that patient, and they can use it from the beginning. So that's sort of where we're headed here. We see that there's we've got basically a parameter space problem, a fitting problem with an optimization problem with higher resolution devices. And what we're demonstrating is that a patient-specific model is potentially a way to solve that. Another thing, we can not just threshold, we can look at improving the visual quality. So we can predict the spatial response of these retinal ganglion cells. And what's shown here is that for an electrode positioned here and within the dotted circle, that you actually don't have the lowest threshold right below the electrode, but adjacent to it. And again, that's indicative of axons of passage going underneath the electrode, it's not shown, but they're going in this direction. And the sodium channel band being the most sensitive point for activation for epiretinal stimulate for a retinal ganglion cell. And you can appreciate that even though you have retinal ganglion cell soma distributed evenly relative to the electrode, you get different responses based on the pattern of the axons. So that's what that shows. So from the patient-specific model, we found that we expect that continued development of this approach will streamline threshold measurement and overall fitting. What we want to do next with this is we want to predict, do multi-channel stimulation and make predictions of the best, most focal perception using current steering approaches. And we hope to validate that in a patient, and we're working towards that goal. It's kind of difficult to bring these patients in but you know, we're working towards that goal. So that's what we are moving. That's the direction we're moving in this work. So to summarize what I've talked about, the first retinal prostheses have received regulatory approval, partially restored vision, so people could see things. People did benefits in limited circumstances for mobility. Since I have a little bit of time, I can give you an example of that. We had two examples. One woman who had the device described how she recognized her the entrance to her apartment building in New York based on where the light was and what the light looked like with her Argus 2. So this gave her confidence that she was in front of her building and not another building. A gentleman in California who took a bus to a certain a building that he would frequently visit. When he got off the bus, he was not always in the same location on the sidewalk, but he was able to find the door for that building based on the vision he perceived with his Argus 2. So there's, again, very specific circumstances, but tangible benefit. More generally, though, many people reported enjoying being visual again. They felt more socially connected, but that's probably not enough to really continue the use of this device. We need to provide more real benefits to people. So we think that electrode arrays need to cover a wider field to provide peripheral vision, but also need to be denser in the center. So we're working on two different technologies that will address those shortcomings in current devices. And we also think there's a role for computational methods to streamline the process of fitting the device to the patient. So this is a group. Maru worked on the electrode array, the wide field array. Elena and Dorsa worked on the intraretinal stimulation, and Kate worked on the computational modeling. So thank you for your attention, and happy to stick around and answer questions even past 9 o'clock if anyone is interested.
Okay, so first let's thank Jim, and unmute yourself, and we can thank you very much for this exciting talk. And if there are any questions, I have a few, but maybe anybody else would like. So maybe I'll begin. Jim, thank you for the exciting talk. I would like to ask you about, so you are showing some kind of an intraretinal approach that could be very helpful, or maybe some kind of an epiretinal approach with a wide field approach. And what are your thoughts about addressing specific retinal ganglion, like what E.J. Chichlinsky is trying to do? What do you think is the importance, and do you have any plans to do something like this with your?
So I think the challenge with E.J.'s approach is the primate retina, is the parafoveal region where you have a group of retinal ganglion cells that are 5 to 7 layers thick. So his providing cellular level stimulation in the periphery may be possible because retinal ganglion cells are arranged in somewhat of a monolayer out in the periphery. I'm not sure exactly when that starts, but as you get closer to the fovea, retinal ganglion cells start stacking up, and then you're no longer going to be able to achieve that resolution. So that's what limits that approach. And so much of our high acuity vision is in the fovea. But the retina itself is challenging in that way, right? So that even if based on Daniel Polanker's paper, where he did an analysis simply based on geometry. So it's really not as much based on anatomical considerations as it is on geometry. But you would need to, even if you can space electrodes 40 microns apart, you still have 20/160 vision, right? So even if you can get electrodes into the retina or against the retina at a resolution of 40 microns between each electrode from center to center, you still have 20/160 vision, which is better than being completely blind, but still it's it's still not, it's still, they would be considered, someone would be considered low vision. And this really motivates the visual cortex prosthesis, just because through cortical magnification, we know that you can use a less dense electrode array and still get near 20/20 vision if you can activate the visual cortex in such a way that allows that. So yeah, EJ's approach is, first of all, his science is fantastic, and his analysis methods are excellent. So I'm not denigrating that work. I'm saying that the practical side of it, the translatable side of it, will run up into and he knows the primate retina very well, so he knows this. And but the multi-layer retinal ganglion cell at the fovea will make that approach challenging.
Okay, thank you. Ellie, you have a question?
Hi, Jim, I am questioning the need for a wide field prosthesis.
Okay.
Especially something as large as you proposing 113 degrees, you said, I think. Where is the evidence? You know, people are classified as legally blind with 20-degree residual field, and we know that they are still doing quite well with 20-degree. It's only when they drop below 10 that they become impaired by or disabled by the impairment. So where are you going from 20 to 113 while this is causing a problem in the insertion and in other things? What drives this?
Right. So there is some evidence for reduced mobility when vision falls below 60 degrees. I can't recall the exact paper that looked at this, but there is a degradation. There was a measurable degradation in that. And so I'm sure you're right that people who have 20 degrees of vision can get around quite well, but it's still, if we want to provide if we want to restore vision, right, or get closer to restore vision, I think we shouldn't ignore peripheral vision.
But I work in peripheral vision and restoring or improving peripheral vision, but it has to have some reason for the big effort that you're putting in. And if you get 20 degrees, there'll be a lot of applause on the way to the 100. So, which is, it's a huge factor. So
there's I don't know if 113 is necessary, but that one thing Daniel found in his Prima, so Palanker's publication in 2022, is that people found it much easier to localize the letters that they were trying to read when they used augmented reality glasses. So this means they could use their natural peripheral vision to kind of align their eyes, right? And then they use the artificial vision to read.
I'm not disputing that having wider field of view is better. This goes without question that 113 is better than 20.
I don't know if 113 is the right number. That's what we did. So we can, I guess it's a demonstration that we can achieve better than what's out there, and it's to be determined what that number should be.
The other question, if I may, since I don't see any other, is where is the evidence that the problem was the resolution with second sight? I mean, almost all that was measured was resolution. Almost all that is measured is resolution. And I parenthetically want to say that if you find a distance between the electrodes and you basically get binary results from the electrode. So to translate that resolution to equivalent of acuity is incorrect, because vision, natural vision, and even impaired vision is with many bits. So it's not equivalent to having a binary resolution image. But But I'm not sure that I've seen any evidence that the problem in seeing with the previous devices had much to do with resolution.
There were multiple issues. One of the major issues that prevented us from really digging into whether or not that was true. So to get everyone to know what they did is they used a grading acuity test. That was a standard test too.
Yes.
That's how they got to visual resolution, is they had a 4 alternative force choice test with grading acuity, and the patients described the direction of the lines. So that's how
Right, but it's
retina
been
implant,
excuse
alpha,
me,
that's how alpha IMS and Argus
Yeah.
and Argus and Second Sight were able to say this is the retinal, this is the visual acuity. Or they used a Landau C optotype. So that's the C of shrinking size, and that has 1 of 4 orientations, which is what the Prima used. So some standard visual acuity tests to get at that. With the Argus, one of the challenges was the instability of the perceptions, is the rapid fading, right? So whereas if you are doing an eye test, you can see, well, you can do the eye test behind me here, right? So you have this eye chart here, right? And behind my head.
Yes,
If you look at that, you see it. You see the E the whole time. With the Argus, most patients would be able to see something, but then it would rapidly fade. So to me, that's one of the major challenges of artificial vision is fading. And the Prima may be able to overcome that, and the NR600 may be able to overcome that by introducing saccadic motion which will simply by that physical motion will vary the electric field and electrical stimulation in a way that perhaps can mimic the functions of saccades and natural vision. But so I agree with you that it's hard to know. It's hard to know why they didn't see well or use the device well. But I know that until we can get until the perceptions are stable and longer lasting than they were, it's hard to really investigate, right?
Yeah, but then people continue to look at those resolution targets, and it's been clearly shown that one can pass that test without having the resolution by using other cues. So it's really not, what I'm saying is the measurements is faulty. The resolution that they got may not be the resolution that the patient had. I don't at all disagree with you that the temporal aspect have been ignored too much because there's a problem how to solve it.
Yeah.
But all of these things don't come up together to say that the effort should go towards increasing resolution. I'm not disputing that having better resolution. I don't know if you see it. I'm seeing my shirt on this screen, and I'm seeing problem with resolution. Do you have the same effect? Dramatic. Maybe it may
I see
be,
what you mean. Yes, I do.
Yeah, you see with so resolution can be a problem, but the nature of it is quite complicated, as this simple image shows. So basically what I'm saying is that we need to understand where the problems are before trying to solve them, and getting larger field than we think is needed is not going to lead to success.
Eli, what do you think are the problems if they are not visual field and
Well, I think the problem is,
acuity?
sure, thanks, Yossi. I think the problem is that we're not testing for vision. We're not trying to understand, we're testing for acuity, for visual field. These are tests that were designed for a working visual system, for the natural visual system, and they are reasonably fit for that task, but they are really not the test that should be used for prosthetic vision. We have to start thinking about what is it that we want from that. And
So, so yeah, Second Sight did develop the Flora assessment, right? So that's a assessment of how people use the devices in the real world. But I think you can do both, right? I mean, we can. It's important to make measurements. It's good to have numbers. It's almost going to be required by any regulatory body that you do these type of clinical measurements, but it's not sufficient. I agree, and it would be. I don't know if the study's been. I don't recall Second Sight doing this study, but I know, interestingly, that anecdotally, that one of the early patients with the Argus used his device 8 hours a day, and this is someone who had almost no visual acuity. So it often goes to the person, you know, how they adapt to using the device, and do they want to use it. There's some patients who had quite good visual acuity who just weren't happy with what the results that they got just functionally, so they didn't use it very much. So yeah, I think we need to do both. We need to make measurements, but we also need to use patient-reported outcomes and assess their how they use the device in the real world. Because people will have an adaptation, and based on their own cognitive abilities, their own determination, they will, find ways to use the device in just the same way that people with low vision accommodate to still function in the world, is just in spite of their loss of vision, visual function.
well, the people with low vision have no option to remove the low vision. The people with a prosthetic can turn off the switch. And so the analogy doesn't work well. But the challenge that you guys are trying to embark is to do better than low vision with macular degeneration is humongous. Better than blindness,
AMD is hard. AMD is very hard.
Yeah, better than blindness is tough enough, as we've seen.
Yeah. So taking on AMD is very challenging because their electronic magnifiers can be used. And then what we saw in the PRIMA trial, and I don't know how prevalent this is overall, is that of course, with an implant, you're going to put it in the worse eye, right?
Well,
but the patient.
No, this is the wrong place to put it. I understand that may be what the FDA wants you to do, but it's wrong. And so the community has to object. This has been a problem with the implanted telescope and other things. You can't put it in the worst eye because you guarantee that you'll fail. If you want to fail, then sure, do it in the wrong eye. No low vision device is being fitted to the worst eye.
But for patient safety, it's absolutely mandatory. How could you do that ethically? How could you ethically do that, Ellie? Come on.
Well,
you can't put a patient at risk of
having their vision get worse. Worse eye, you guarantee a worse output for the outcome for the patient. So you're not protecting the patient.
That's a good point. That's right. So it's there's the challenge, right? So you have this. But you have a mandate
not to abide by the regulatory, but to fight them. To do.
How is that going? How's your fight with the regulatory agencies?
I fought with them a couple of times, and usually I succeed because
that's good.
They don't really have all the knowledge, and that's why they have expert committees and other structures to address this. So
it's we should push back. You're right.
Yeah, yes.
It's not
on this point we should push back very hard because
there was this conference of the FDA, sorry, and Ellie, maybe you should fight with them.
Well, I try, whenever I have a chance. I actually enjoy fighting with them, so there's an extra benefit.
Okay,
But I think it's the right thing to do, and we need to look at what's the right thing to do and push for that.
As long as, you know, with informed consent and the patient knows the risks, right? And you can explain to them and justify that the benefit you will get is if we put this in your better eye, but the risk is greater. So the bigger benefit, bigger risk.
Right. But it's well known and clear and simple to explain why putting it in the worse eye will not result in a positive outcome for the patient. And this is what it's about. It's not about for safety trials or things like that. You could put them in the worse eye to see that, you know, it works as safety is okay to do this. But once you move to a efficacy, even before effectiveness, even efficacy is not reasonable with the worse eye.
Eli, I think that we
It's a
should
tough-
try maybe to thank Jim, and if you want, we can stay here and have a.
No, it's okay.
So
actually-
let's. So there are a few brave people who stayed late. So thank you again, Jim.
Thank you for having me. Enjoyed the conversation and the discussion afterwards.
And we are looking to see you maybe in the Arvo or other things. We can stay here in this room if you want with Eli, and we can discuss it if anyone wants.
So I would like to hear about the telescope. Have you been involved? What's I know that it exists, the intraocular
Yeah.
telescope.
I've
How
been
is the-
involved